Hello everyone, welcome to Care Microbiology classes. So TS set notification వచ్చేసింది కదండి. You all are very eager to crack the TS set. Those who are aiming for the degree lecturer jobs, so TS set is very important. So syllabus చూస్తే మనకి TS set syllabus is almost all same to the net syllabus. And first of all, I'm conveying my best wishes to you. మీరు ఖచ్చితంగా TS set qualify అవ్వాలని నేను కోరుకుంటున్నాను. And in this TS set for the life science stream, I'm making some videos which might be useful to you. In that one, for the biomolecules regarding the carbohydrates, I'm making this video. So in this video, I'm going to discuss about definition, classification, and monosaccharides anatomy and stereochemistry. A little part of the stereochemistry, I'm making this video because if I make a long video, that may not grab your attention. So that's why I'm making small videos. Okay. So what is the carbohydrate? So carbohydrates general definition, we all know that they are the polyhydroxyaldehydes or polyhydroxyketones and the compounds which can produce them on hydrolysis. All these are called as the carbohydrates and its general formula is CH2O taken N, right? And there are some other common names are also used they are also called as saccharides, they are also called as glycans and glycomes or sugars. Especially we use the term sugar when the carbohydrate is sweet in taste and they can soluble in the water. We may call it as a sugar, okay? And simply if we see the etymology of the carbohydrates, these are the hydrates of carbon, simply. Okay, and how much energy the carbohydrates gives, because carbohydrates are the very primary source for all the living organisms for the energy, right? So in this regard, 1 gram of glucose upon oxidation, it gives 16.7 grams, 16.7 kilojoules of energy, or we may call it as 4 kilocalories per gram. Okay, that is the energy. And elsewhere where it is useful, ఇంకెక్కడెక్కడ ఉపయోగపడుతుంది కార్బోహైడ్రేట్ అని అంటే, so for giving the structure of many living things, like for the bacteria, for the plants, and for the insects, for their cell walls and the exoskeleton, it is useful. I gave here. So cellulose in the plant cell walls and chitin in the insects and peptidoglycan it is in the bacterial cell wall. So you may get, if the question is very easy, they will ask that which of the following is not a carbohydrate or something like that. So you must know where the carbohydrate was there, right? And another one is if we see in cells, in living cells, cell to cell recognition is also because of the carbohydrates, such as glycoproteins and glycolipids participates in the cell adhesion, immune responses, and blood group antigen determination. So for the ABO blood groups, antigen is very important, right? So in the name of glycoproteins, they play this vital role. And if you see in the metabolic activities, so these ribose, deoxyribose, right, they are the very important for any living organism. So in that ribose and deoxyribose, that particular ribose is nothing but a sugar, we all know that, right? And in the ATP, ADP, AMP, NAD plus, NADP plus, FAD, coenzyme A, in all these compounds, sugar is there. I mean, carbohydrates are there. So moving to the next slide, history of the carbohydrates. So in many books there is only like carbohydrates, monosaccharides, polysaccharides, disaccharides. Itlane undandi. So I want to tell you about history of carbohydrates. So if we see here, in 1747, first time this sugar is obtained from the beetroot, and in 1812, Kirchhoff showed that the acid can break the starch, and that starch can be break down into the glucose using some acid. And in 1844, the German scientist Carl Smith, he coined the term carbohydrate. He coined the term carbohydrate, and in 1891, Fisher mapped that he mapped the 3D structure, 3D shape of the glucose, 3D shape of the sugar, and he won Nobel Prize in 1902. And in 1937, uh, in 1937, Walter Haworth, he confirmed that the sugar flows into the rings, and for this invention he got a Nobel Prize in 1937. And in 1947, uh, in 1947, it was also observed that this sugar also plays major role in the metabolism. And coming to the next slide, so how these sugars or carbohydrates are classified? These carbohydrates are classified based upon three things. Okay, the first classification is depending upon the number of units present, or by size we may call it as. And the second classification is depending upon the composition of that particular carbohydrate. And the third one is whether it is reducing or non-reducing. Depending upon that chemical property, they were divided. Okay, so let us see. First one is the depending upon number of units. Okay, so depending upon the number of single carbohydrate unit was present, they were divided into three types. One is the monosaccharide, second one is the oligosaccharide, and third one is the polysaccharide. Here we have to remind that saccharide, it is a Greek word, means sugar. Okay, so when we hydrolyze any one sugar compound or carbohydrate, so it cannot be hydrolyzed further. Means if you see that if we hydrolyze one polysaccharide, it can be hydrolyzed, it can be lysed into the two monosaccharides. Okay, but if you హైడ్రోలైజ్ ఐ మీన్ దాని కనుక మళ్ళీ మనము హైడ్రోలైజ్ చేస్తే మోనోసాకరైడ్ ఇంకా అంతకంటే విడిపోదు అనమాట, ఇంకా అంతకంటే విడిపోవడానికి లేదు, ఇంకా అది అలానే ఉండిపోతుంది. సో వాట్ ఎవర్ ద one carbohydrate which cannot be further hydrolyzed, that is called as the monosaccharide. And when a compound can be hydrolyzed into the 2 to 10 units, then it is called as the oligosaccharide. And if that compound can be hydrolyzed into more than 10 units, then it is called as the polysaccharides. Okay, so here if we go to deep down the examples of the monosaccharides, oligosaccharides, and polysaccharides, it is much more deeper knowledge and also important. So I gave here some one or two examples. Okay, so monosaccharides, very important and very well known example is glucose, fructose, ribose. And for the oligosaccharides, it is maltose and sucrose. And for the polysaccharides, starch, glycogen, and cellulose. These are some of the examples. Okay, and the second classification depending upon the composition. So if any one particular compound or complex compound which was made up of single type of monosaccharides, so single type of monosaccharides together formed a chain of polysaccharides, then it is called as the homopolysaccharides. The best example is cellulose, it is made up of glucose unit. Okay, and heteropolysaccharides. Heteropolysaccharides is the mixture of monosaccharides and their derivatives. Means here along with the monosaccharides, means not a single type, there may be different types of monosaccharides, and along with those different monosaccharides there might be the derivatives of the carbohydrates, means like amino acids like that. I gave already here, see these. The best example for this heteropolysaccharides generally called as a GAG. Okay, glycose, aminoglycans, and amino sugars and uronic acids. So these together called as the GAGs. Okay, and you have to get the question GAGs ante అవి ఎక్కడ యూజ్ అవుతాయి అని. I will explain it in the next video. Okay, అండ్ ఇఫ్ మీకు తెలిస్తే ఖచ్చితంగా మెన్షన్ చేయండి కామెంట్ సెక్షన్లో. అండ్ ద థర్డ్ కంపోజిషన్, ఐ మీన్ థర్డ్ క్లాసిఫికేషన్ దేని మీద ఆధారపడి జరిగింది మీన్ అంటే సో ద థర్డ్ క్లాసిఫికేషన్ డిపెండింగ్ అపాన్ ద రెడ్యూసింగ్ ఆర్ నాన్ రెడ్యూసింగ్ ప్రాపర్టీ. ఇఫ్ ఏ షుగర్ ఈజ్ రెడ్యూసింగ్ when it retains a free anomeric carbon, then it is called as a reducing sugar, otherwise it may call as a non-reducing sugar. Okay, so here I am going to explain you about simple sugars or monosaccharides. I am going to explain you. So monosaccharides usually based upon the functional groups, there are two types. Okay, and based upon the number of carbons, it is I gave it here 3, 4, 5, 6, 7 carbons వరకు ఇక్కడ మీకు ఇచ్చానండి. Okay, so depending upon the number of carbons is one classification, and depending upon the functional group there is another classification. Okay, so aldose, if there is terminal aldehyde group, then it is called as the aldose, and if it is the terminal ketone group is there, then it is called as the ketose. ఓకే. సో ఇక్కడ చూడండి ఆల్డోస్ కి కీటోస్ కి కెమికల్ ఫార్ములా సేమ్ గా ఉంటుంది. గ్లిజరాల్డిహైడ్ అండ్ డైహైడ్రాక్సీ ఎసిటోన్ యూజువల్లీ కాల్డ్ as the DHA. For the glyceraldehyde and DHA, the chemical formula is same, but depending upon the presence of CHO or C double bond O, difference is there. Okay, and next one is the glucose and fructose. So if it is the aldehyde group, then we will call it as the glucose, and if it is the ketone group, we will call it as the fructose. And depending upon the number of carbons, see here, if it is 3 carbons, we will call it as the triose, and 4 tetrose, 5 pentose, 6 hexose, and 7 heptose. And we also gave the names here, right? Triose, glyceraldehyde or DHA, tetrose, erythrose, pentose, ribose or ribulose, hexose, glucose or fructose, heptose, pseudoheptulose, right? And what is their significance? That one also I gave here. And here you have to definitely remember some chemical properties of these things. So among that, first one is the chiral carbon or chiral carbon. You can pronounce anything. So when I was a student, I was a degree student, I got so many thousands of doubts about these things. So I will try now to give as much as explanation that may helpful to you. So what is the chiral center? So sorry for my pronunciation if it is wrong. You just ignore it. Okay. So what is the chiral carbon or chiral center? So a carbon which was bonded to 4 different groups, that carbon is known as the chiral carbon. Okay, so here once again I'm particularly telling you which carbon is connected to 4 different types of groups. That particular carbon is known as the chiral carbon, or we may call it as the asymmetric carbon. Remind this. Okay, chiral carbon or asymmetric carbon, and that is known as the chiral center. Okay, and next one is the 2n rule. So depending upon the how many chiral centers are there, that stereoisomers number is dependent on the number of chiral carbons. Okay, so here n is the chiral centers and 2n is the stereoisomers. So ikkada aldo hexose ki chuste manaki we have 4 chiral carbons. We have here that figure also, see here. So below the CHO, here one is there, no? 1, 2, 3, 4. So total 4 carbons are there, right? So these 4 carbons have the 4 different uh, groups, right? So 2 power 4. So the one aldohexose may have 4 to 16 isomers. They may be 8 are D and 8 are L. Okay. So here we have to get another doubt. What is D and what is L? I gave here capital D, capital L and small d, small l, right? So here there is some differentiation in each textbook, but you can remember it in either way. There is no problem. Okay. So first one is the capital D and capital L. Okay. So this is regarding the H and OH arrangement, which was next to the primary alcohol carbon. బాగా గుర్తుపెట్టుకోండి. సో ఐ యూస్ టు టెల్ ఇన్ తెలుగు. ఓకే, ఇక్కడ మీకు ఇప్పుడు మీరు ఈ స్ట్రక్చర్ చూస్తే మనకి ఆల్కహాల్ గ్రూప్ ఎక్కడుందండి? చివర్లో ఉంది కదా, CH2OH. దీనికి నెక్స్ట్ ఉన్న కార్బన్ కి దానికి ఇటుపక్క అటుపక్క ఉన్నటువంటి హైడ్రోజన్ అండ్ OH, ఈ రెండు ఎక్కడెక్కడ అరేంజ్ అయి ఉన్నాయి అనే దాన్ని బట్టి దాన్ని we may call it as D and or L. So if the OH group is on right side, we will call it as the D compound, or if the OH is on the left side, we can call it as the L compound. Okay, so this is the first one. Second one is the small d and small l. So capital D, capital L is about the configuration. Please remind this. Capital D and capital L is about the configuration of the OH and its spatial arrangement. And whereas coming to the small d and small l, it is regarding its dextro rotation or levo rotation. In the presence of polarized light, how it can rotate. If it rotates to the right side, it is called as the dextrorotatory, and if it rotates to the left side, it is called as the levorotatory. We can write it like small d small l or plus or minus. Okay. And this is what all I want to tell you in this video. And in the coming video, I'm going to explain you about epimers, enantiomers, stereoisomers, anomers, and mutarotation. So if I missed anything, so please mention so that I will explain about those things in the next video. Thank you so much.